My name is Marian Pitters, and I'm delighted to be the facilitator for today's webinar. I'm here to introduce our expert speakers, moderate some questions from you, our participants, and ensure that the webinars progress according to the schedule so that we respect everyone's time. But first, on behalf of McMaster University, it is my pleasure to welcome you to the fourth webinar of the 10-part series, Expert Perspectives on Pandemics. We have participants today from all over Ontario, Canada, and the globe online. Graduate students, faculty from McMaster and partner institutions, as well as representatives from the Pan American Health Organization, PAHO, the World Health Organization, WHO, and some select invitees. Many of you are preparing to contribute your skills to mitigation of the COVID-19 pandemic. We welcome you all to today's compelling session. The COVID-19 emergency presents a global challenge to our healthcare systems, our economy, and our ways of living. In the face of this unprecedented global health crisis, McMaster is dedicated to innovation and collaboration in the discovery of solutions to help combat COVID-19. This series is integral to the Master of Science in Global Health. Experts will delve into topics including historical trends, vaccine development, the threat of antibiotic resistance, and Canada's research response to COVID-19. A quick overview of the 10 webinars in our series can be found at the link where you registered, globalhealth.master.ca/2020 webinar series. Today's session is webinar 4, Antibiotics, Resistance, and Pandemics. It features Dr. Jerry Wright's perspective on the complexities of antibiotics, resistance, and pandemics. A final housekeeping note before we start. Time has been built in for questions and discussion at the end of the webinar. Please use the chat tool to submit questions electronically. And please note that while we may not be able to get to all of your questions, they will be forwarded to our speakers, and we will aim to address them afterwards on the Global Health website. At this point, I would like to introduce Dr. Andrea Baumann. Andrea is the Associate Vice President of Global Health, Faculty of Health Sciences at McMaster University, Director of the Master of Science in Global Health program, and the Director of the World Health Organization, WHO, Collaborating Center in Primary Care and Health Human Resources. In addition to her many awards and accolades, in 2018, Andrea's groundbreaking research and outstanding nursing leadership were recognized with her appointment to the Order of Canada, one of our country's highest honors. Andrea, will you please start us off with a few words about launching this webinar series?
Thank you, Miriam. I'm very pleased to offer the opening remarks on behalf of Dr. O'Byrne, the Dean, Vice President, Faculty of Health Sciences at our Michael D. DeGroote School of Medicine. He introduced the first inaugural lecture, and I know he wants me to continue to welcome our participants from this week as well as last week and any new participants joining us today. It goes without saying that we have challenging times. We organized the speaker series to provide an overview that would give everyone listening a transdisciplinary viewpoint of the nature of the pandemic. We have tried to cover many different aspects, but we would like to hear from you near the end of these WebExes as to what you would think, how we could further on this series. I definitely want to thank Jerry and previous speakers for sharing their expert perspectives on pandemics and issues related to pandemics. I'd like to thank the students for joining us today and the faculty and partners in Global Health who are with us today. It's been our privilege to organize this groundbreaking webinar series for all of you. Thank you.
Thanks, Dr. Baumann. Before I introduce our speaker, a quick reminder to post your questions throughout the presentation in the chat box. Dr. Jerry Wright is the Canada Research Chair in Antibiotic Biochemistry, the Director of the DeGroote Institute for Infectious Disease Research, and a professor in the Department of Biochemistry and Biomedical Sciences at McMaster University. Dr. Wright's research is focused on antimicrobial discovery with an interest on natural products as sources of new antifungal and antibacterial agents. He co-founded the Canadian Anti-Infective Innovation Network and is also a Fellow of the Royal Society of Canada. When not thinking about infectious disease, Jerry plays drums in a faculty rock and roll cover band called the Null Hypothesis. Dr. Wright, we look forward to your perspective on the antibiotics, resistance, and pandemics for 30 minutes. I'll provide you with a reminder at the 28-minute mark, and then we'll invite you to engage our global participants today in a Q&A session for 15 minutes. Over to you.
Well, thank you very much, Marion and Dr. Bauman, for allowing me to give you some of my thoughts on how antibiotic resistance intersects with the COVID issue and pandemics in general. So just want to confirm that you can see my slides. Let's take that as a yes. Okay, so again, thanks very much for giving me the opportunity to speak. And what I want to do in this, okay, this is now giving me grief. Here we go. Now I know how to do it. What I want to do is really touch on three things today. Sort of go over what antibiotics are and what's their impact on human health. What is antibiotic resistance? Where does it come from? And how does antibiotic resistance intersect with this crisis that we're all living through? So first, what are antibiotics? So it's important to sort of ground this discussion in an idea or in some history. And of course, before we had antibiotics, we really didn't understand, or at least until the 19th century, we didn't understand what the source of infection was. We understood pandemics, we understood epidemics and outbreaks. The whole history of humanity is riddled with these, and it's in our literature and in our art. But we really did not understand what caused it. The world changed in the 17th century with the discovery of this absolutely groundbreaking instrument, which you all recognize as a microscope. So that all of a sudden, for the first time in the history of humanity, we were able to see things that were not visible to the naked eye. And that transformed our understanding of disease, eventually connecting these organisms that we could see that were tiny and could not be seen with normally with diseases. And that was just if you think about the history of medicine, the understanding of infection caused by organisms that are not visible in normal life was absolutely transformative. Once we understood that, though, with leaders like Pasteur and Koch, the question was then, is there anything we can do about these microbes? And the answer, of course, came with antibiotics. And so early in the 20th century, we saw the discovery of this remarkable molecule that you will recognize as penicillin. And one of the points I like to make about this discovery was it took a long time for the initial discovery of the activity of penicillin from coming from a mold that was discovered in the environment by Alexander Fleming on the left of this slide, who was a microbiologist working in London. He couldn't really do anything with this discovery for years because he simply did not have the tools to be able to do so. You had to bring together a whole team. So Ernst Chain in the center is a chemist who helped isolate and understand the structure of penicillin. Florey was a pathologist who could actually do clinical trials and see patients. And Dorothy Hodgkin was a biophysicist who actually was able to solve the structure of penicillin using X-ray crystallography. So this is really an important take-home that is applicable not only to the antibiotics, but also to the current crisis that we're in with COVID, and that is a single individual is not going to be able to solve this problem. It's going to take scientists from many, many disciplines to come together to be able to solve these kinds of problems. And the roadmap for that was established in the antibiotic era. So this all occurred, the discovery, Fleming's discovery was in 1928, but it really took the Second World War, the early 1940s, for it all to come together so that we could actually have penicillin as a drug. And once that discovery happened, that is the understanding that microbes from the environment might be sources of drugs to battle the infections that we have been dealing with for millennia, it rapidly exploded. And so we saw the discovery of the first drug that was useful against tuberculosis by Selman Waksman and Albert Schatz, streptomycin, tetracycline, an antibiotic, had excellent activity against a lot of gram-negative bacteria by Ben Dugar, cephalosporins, which took beta-lactam class like penicillins into a completely new area by Giuseppe Brozzu, and then again another milestone discovery in particular applicable to tuberculosis in the Le Petit laboratories in Italy by Piero Sensi. And this is just a few examples of what we now call the golden age of antibiotics, basically from 1945 to 1965, when virtually all the classes of antibiotics that we have come to know and use in the clinics today were discovered. And I'll touch on that in a second as well. And the impact of antibiotics has really been profound, right? So, and one of the ways I like to frame it is it actually has changed the way that we die. So if you think of the pre-antibiotic era, this is data from Statistics Canada that I pulled from 1920 after the first wave of Spanish influenza, you will see that that what was killing people were infections, so diseases of early infancy, tuberculosis, pneumonias, gastritis, communicable diseases as a whole, and cardiovascular disease and cancer were important, but together these infectious diseases were responsible for well over half of deaths. Flash forward to 2009, this has completely changed. It's really cardiovascular disease, stroke, cancer, diabetes, Alzheimer's. So what's changed now is that we've controlled infection with things like vaccines and in particular antibiotics, and changed the way that we die. So we now die in old age of many chronic diseases, and fewer people die, at least in North America and Europe and other areas, similar areas, where infection is now taken a backseat to that. So antibiotics, though, it's important to realize that not only do they have an important impact on infectious disease, they also freed up the medical sector to be able to do a number of different things that were completely unthinkable when infection was a concern. So we can do major surgeries now, heart surgery. Imagine that without proper infection control given to us by antibiotics. As we get older, we oftentimes are starting to wear down, and so hip and knee replacements are very popular. Obviously, cancer chemotherapy, which ruins the immune system oftentimes during treatment, results in a series of secondary infections that are controlled by antibiotics. Organ transplantation, similarly, with a suppressed immune system. And of course, preterm infants, who often in the past never would have made it grown into adults, are now taken care of with antibiotics. So without antibiotics, we lose all of these things. So it's not just control of infection, it's really modern medicine the way we know it. So this is my timeline, or my history of antibiotics by Jerry. So we start out, we're all happy we have a new drug. Over time, we lose this drug because of resistance. But that's okay, because we can just repeat this cycle and find new drugs. And in fact, this has been the history of medicine post the 1940s, when we discovered the first drug, penicillin. This cycle has occurred over and over again. And the concern about antibiotic resistance, while always there for the quote-unquote professionals in the field, the clinicians and drug discoverers who knew that resistance was always an issue, but we could always find a new drug. We're now in a situation where that cycle is broken, where resistance continues apace, but the discovery of new drugs is turning out to be much more difficult than we want. So you can really see this nicely in this timeline where, as I mentioned, we have this golden era of antibiotics between the 1940s and the 1950s, a few more in the 70s, and then nothing after the mid-1980s. So no new antibiotic classes have been discovered. We've had derivatives or versions of some of these other drugs, but nothing brand new. And that's a major cause of concern for us. This indeed is the antibiotic resistance crisis right there. So why is this happening? It's all this guy's fault. This is really evolution by natural selection. So antibiotics are toxic molecules to bacteria. They provide a very strong evolutionary pressure to be able to escape and live another day. Because bacteria divide so quickly, the generation time can be counted oftentimes in minutes rather than decades like us. You can sample a lot of genetic diversity very, very quickly. The other thing that bacteria are really good at doing, not only in just simply evolving de novo, but they actually exchange genetic material very easily. So they have sex with each other on a global scale and exchange resistance genes often, as you can see, from one organism to another. This long connection is called a pilus, and that pilus transfers DNA from one organism to another. And they don't have to be related, these bacteria. They can be very, very different species and even different genera. So the AMR crisis is fundamentally due to evolution and the behavior of bacteria, but it's also due to our behavior. So we live in a global society, and of course, we're really starting to realize this during the COVID pandemic, how interconnected our supply lines are across the globe. So we can travel either people or goods very, very easily. At least we could up until a few months ago. And of course, all of these have are these entities, whether it's people or animals or food, come across with bacteria on them. We have large-scale production of food to feed a population of humans that the planet has never seen before. North of 6 billion people are on the planet, all of whom need protein. That causes intense farming practices, and the use of antibiotics in farming has been absolutely dwarfing the amount of antibiotics that we use in human health, actually, by almost a factor of 10. And of course, because of all these people on the planet, we have a tremendous amount of environmental degradation. And this is causing a lot of mixing of organisms that used to be separated from the past. And a lot of emerging pathogens in bacteria are environmental pathogens, and they come with a lot of new resistance genes. And of course, we've seen this with the COVID pandemic as well, where because of encroachment of humans on ecosystems that we used to not interact with in a significant way, and we've been exposed to viruses that otherwise would have been pretty rare. So there are microbial reasons why this is happening. The AMR crisis is happening. There's human reasons why it's happening. Of course, there's economic reasons why it's happening as well. And this is really reflective of the challenge that the for-profit drug companies are having in making money on new antibiotics. And you can see this with this is just some data for the past 5 years from NASDAQ. All of these are small drug companies that have taken up the antibiotic discovery mantle after the large pharmaceutical companies have left it. And you can see that despite the fact that all of these companies, except for Entasis, which just came up a couple of years ago, have drugs, they did it. They actually discovered new drugs to help us. Half have either gone bankrupt or have been forced into mergers where their assets have been diluted. So it's a really significant challenge to bring people into this field to be able to find new antibiotics. So where does this resistance come from in the first place? Well, antibiotics, of course, work by blocking essential bacterial physiology, DNA synthesis, membrane proteins, the synthesis of proteins, the integrity of membranes in the cell wall. And of course, bacteria have many countermeasures. So they can degrade antibiotics, they can change their structures, they can just simply mutate over time, as I indicated before, or they can pump them right out of the cells using a series of very sophisticated biochemical machines, often with very broad specificity. So one pump can pump out many different classes of antibiotics. And it's important to realize that this resistance is, in fact, a pandemic. So this is a global in nature. This is data I took from the Resistance Map site. You can find here on the slide. This is resistance of E. coli to fluoroquinolone antibiotics. The darker the blue or gray color here, the more resistance there is. You can see it's all across the world. There's some more examples. Third-generation cephalosporins to Klebsiella pneumoniae, Acinetobacter, carbapenem antibiotics, vancomycin resistance, and enterococci, and MRSA or MRSA that I think many of you have probably heard of before, at least not in your personal life, than in the news. So it's everywhere, and it hits all sorts of different kinds of classes of antibiotics and microorganisms. So that suggested to us several years ago that because this is spread so quickly and it seems to emerge to all antibiotics, that there must be a deep reservoir somewhere of resistance genes. And so several years ago, my lab decided to think about this in a different way and said, well, maybe the environment, the bacteria that live in the environment might be the source. And so a graduate student at the time, Vanessa DiCosta, and her colleague, an undergrad, Kate McGran, went out into a number of environments in Ontario and isolated bacteria from the soil. This is just an example of these. And they isolated 500 different bacteria. And what did they find is that they were all drug resistant. And in fact, they weren't just resistant to one different antibiotic. They, on average, were resistant to somewhere between 7 and 8 different antibiotics. So if this was an organism that caused the disease in our hospital, we would definitely call it a superbug. And so the environment basically is chock full of superbugs. And that was quite surprising to us. And not only is it the environment that we see today, we actually went into the Lechuguilla cave in New Mexico with my colleague Hazel Barton, who's a cave microbiologist. And she isolated bacteria that live on the surface of this cave. This cave has been isolated from the surface for 3 million years. And we found the same results. That is, all of the bacteria in this cave were multi-drug resistant. So that suggested that this is hardwired into microbial populations across the globe. But it is a contemporary phenomenon. And it suggested obviously not. Maybe it's quite old. And you've already seen this guy, Dr. Hendrik Poinar, who had in a collaboration between Dr. Poinar and my lab, and of course you know that Dr. Poinar is an expert in ancient DNA. We said, where can we find really old environmental DNA? And the answer, at least in Canada, is in the permafrost. So these are ancient sediments in the Yukon and in the territories, northern territories of Canada, that are permanently frozen sediments. And these can be dated back hundreds of thousands of years. And so what we did, working with our colleagues at the University of Alberta, Dwayne Fries, and the Yukon government, Grant Zazula, we were able to bring some of these sediments to McMaster, isolate DNA, ancient DNA from 30,000 years ago, and found, of course, DNA for things you would expect. We found mammoth DNA, and we found ancient bison DNA, and this extinct horse DNA. We also found DNA for genes encoding resistance to tetracyclines, to penicillins, and to vancomycin. This is one of these drug of last resorts that we find. So resistance is really part of the ecology of the planet. And if you start to think about it on a global scale, there's 10 to the 30 bacteria on the planet. It's a stunning number. There's 10 to 21 stars in the universe. There's 9 orders of magnitude more bacteria than there are stars in the universe. There's 10 to the 17 seconds since the Big Bang. Now, these numbers are staggering. All these bacteria have genomes. All of these bacteria have the potential for resistance. And the history goes back billions of years. So resistance is a natural phenomenon within bacteria. It can move between bacteria, as I showed you before. And of course, the selection pressure is the human use of antibiotics, so that these genes start to move from environmental bacteria into the bacteria that cause disease. And the more antibiotics we use, the more the chances of those are of happening. And right now, what the end result is we have bacteria in our clinics that are resistant to all antibiotics that we have. So that's where we're at. It's a really significant challenge. It's a really difficult situation to be in, but it's driven by natural selection. It's driven by evolution, and it's exacerbated by human activity and exacerbated, of course, by the economic challenge of bringing new antibiotics to market in this in global environment. Okay, how does this intersect with COVID? Actually, in a number of different ways. So I'll walk you through here. So, I put in the wrong slide here, but the COVID crisis is obviously, as we all know, this is caused by a virus. It's not a bacteria. So antibiotics do not work on COVID. Because of as I mentioned before, environmental degradation of human populations with natural populations that otherwise would have been rare or even not even happening in the past, we have this mixing of strains of virus with human populations like we've never had before. And the fact that we've seen a new coronavirus emerge in every decade of the 21st century tells you that it's a really significant event. So what you all know is that this is an official pandemic. You can see the red dots here from the Johns Hopkins University hub, really been monitoring this over the crisis. And of course, like antibiotic resistant bacteria, this virus is resistant to pretty much all the antiviral drugs that we have. So it comes from the environment without any sensitivity to existing drugs. If there would have been sensitivity to existing drugs, we would have been using them already, and this crisis would have been dampened in a significant way. So right now, there's well over 3 million cases, well over 200,000 deaths as a result of this. So this is a really significant challenge, as we all know. So is there a link between this crisis and the previous one that I told you about, which is drug resistance? There is, I think, in at least 3 different ways. One is secondary infections. The second is the inappropriate, I think, use of antibiotics. And the third is a potential one for large-scale use of antiseptics. I'll pause here for a second and just say that these are hypotheses based on my understanding of drug resistance. We haven't proven a lot of these connections yet, and there's a lot of science to be done to be able to do that. But this is my view of the world. So first, secondary infections. So what we know is that from the previous global pandemic that we suffered through, which was the 1980 influenza pandemic, most of the people who died from that, and there were millions, of course, seem to have died not from influenza itself, but through infections that set in the lungs after the initial viral attack. We're seeing a lot of this kinds of patterns happening in patients who are hospitalized with COVID-19, and almost all of them, if you are hospitalized, will receive an antibiotic. We did not have antibiotics, of course, in 1918. And this is oftentimes preventive. We want to do is make sure that not get those infections. We know that these kinds of secondary infections are very common in other viral respiratory diseases, and so this is just wise clinical practice at this time. Of course, what this means is that we're using a lot more antibiotics than we would have in the past, and there's a significant opportunity here for the contribution to AMR. The other one is what I would call sort of dubious use of antibiotics, and we have all seen the news reports about the use of hydroxychloroquine with azithromycin, which has been touted as a potential treatment for COVID. Right now, there's on the clinical trials DB from the FDA in the U.S. There's over 50 clinical trials that include azithromycin. Azithromycin is a macrolide antibiotic that has activity against bacteria. It has no activity against viruses. And nevertheless, it's being explored as part of this treatment regimen. There may be sound reasons for that, but the end result is, of course, what we're having now is because everyone's jumping on the bandwagon. You can get azithromycin or Z-Pak, as it's known, from the pharmacy with a prescription, and many people are going out and self-medicating as a result. And not surprisingly, this is an absolutely essential drug, WHO essential drug, and now there are global shortages around the world because it's being used without evidence that it has any effect on COVID. And of course, if you're using lots and lots of antibiotics around the globe, you're creating that pressure for resistance to emerge and to get embedded in bacterial populations. The other element of this is we've all seen these videos of the disinfecting streets and shops, and now they're doing it on a sort of industrial scale, using all manner of disinfectants, which the virus is very, very. But many disinfectants also select for antibiotic resistance. Some of them don't. Things like the alcohol hand sanitizers that you're used to walking in the store. You might have some at home. These do not select for drug resistance, but several of them do. So if you're now thinking about this use of antiseptics like in this way across the globe and in such a significant fashion, there's a great potential to increase drug resistance. So I'm almost done here. The counter argument to all of this is that because we're doing a pretty good job in most places anyways of social distancing, that means we're not interacting with each other, which is how we get a lot of bacterial infections. We're staying at home, we're not interacting with the rest of the world as much as we used to. We're not getting on airplanes, we're not getting on crowded subway trains. We're not going to visit our family members or friends in hospitals. There's a lot less potential for infection. And so maybe, on the other hand, this might actually decrease the pressures for AMR. And as I said before, these are all just ideas, and only time will tell which one will be the most prevalent. So just to summarize what I've said, so AMR, or drug resistance, antibiotic resistance, is a slow-moving pandemic. It's the analogy to climate change has been made several times. This is something that's happening around the world. It's impacted by global transport. It's impacted by human biology. It's impacted, or human society. It's impacted by bacterial biology. It's impacted by economics. But it's moving much slower than, of course, this virus that we're dealing with now. But it's nevertheless still a significant problem that we have to address. And as I mentioned before, it really is an existential threat to the way we practice modern medicine. If we don't have good antibiotics to control infection and to treat infections, we can't think of using all of the amazing technologies that we have developed over the last 60 years to extend our lifespan on average by 20 to 25 years. This threat is just the result of evolution, as I mentioned, and of course the fact that we're not able to bring new drugs to market right now. This infection, or this COVID pandemic, has the potential to spread AMR over the long term because of all the reasons I mentioned. But perhaps in the short term, it might actually have a dampening effect because we're not seeing as many bacterial infections in the larger population, not necessarily the folks who have been infected or in hospital, but in the larger population, we're just not interacting with each other as much. So that's where I'll stop. If you have any questions, I'm very, very happy to take them. Thanks again for the invitation to share these ideas with you.
Thank you so much, Jerry. That was quite a wonderful presentation. Thank you. Everyone, be sure to submit your questions in the chat section. Jerry, we've got a few coming in already, so let me get started on those.
Sure.
We won't even give you a chance to breathe before you have to do this. The first one, what role, if any, does the competition between pathogenic bacteria and more beneficial benign bacteria play in combating infection and/or resistance? So I'll give you that again so the participants and you can have a chance to think about it. What role, if any, does the competition between pathogenic bacteria and more beneficial benign bacteria play in combating infection and/or resistance?
That's an excellent question, actually, and I can answer it in a couple of ways because there's sort of, like everything, there's two sides to it. So on the one hand, bacteria that don't cause disease. So let's think of just a normal human microbiome, the gut microbiome, for example. When you take an antibiotic, there's a significant impact on that. That's one of the reasons why almost every antibiotic has in its little sheet that you get with it that one of the side effects will be diarrhea or gastrointestinal discomfort. It's because you're wiping out those bugs, and a lot of them are susceptible. Of course, they're just bacteria, and they're susceptible to all of the evolutionary pressures that bacteria are. And so, in particular, with chronic use of antibiotics, you can begin to select for your microbiome to be quite resistant to those antibiotics. And then they become a reservoir for those resistance genes that may transfer to pathogenic bacteria. And we've seen instances of this in the clinic and in the lab where dosing of patients with antibiotics results in a very quick increase in resistance, not just in pathogens, but in benign bacteria. And of course, then they become possible sources of resistance genes. The other way to think about it is actually kind of clever, and there have been some efforts to use probiotics, so benign bacteria that have been sort of pre-selected not to have resistance genes. that basically flush out pockets of chronic resistant organisms in particular. And jury's still out as to whether or not that's a really effective way to do it, but there is a potential application here. If we can have those benign bacteria outcompete the resistant ones, if you will. And as I said, that still hasn't become a clinical practice yet, but there have been several studies.
Thank you for that. Another one on effects. If the azithromycin has no effects on COVID, why do we observe an amelioration of symptoms in affected patients when using it? So let me give you that one again. If the azithromycin has no effects on COVID, why do we observe an amelioration of symptoms in affected patients when using it?
That's a really good question, and I honestly don't have the real answer. There's been several thoughts. One is that you're going to get better anyways, and it's just chance. The other is that azithromycin is part of a larger family of antibiotics called macrolides, and macrolides have been known since their discovery in the 1950s to have anti-inflammatory effects. And in fact, there's some macrolide that don't have antibiotic activity, but retain these anti-inflammatory properties. And we know from COVID patients that inflammation, in particular in the airways and in the lungs, is a significant component to the disease. And if you can dampen that, then you're much better off. So that could be that's one of the reasons for that. Of course, there are much better anti-inflammatory agents than azithromycin. And so if that's the mechanism, then I think there are in the clinical trials right now using things like steroids, which are great immune suppressants and anti-inflammatory agents to achieve that benefit, if that's the benefit for azithromycin. But again, I think if what I have seen so far of some of the studies, the amount of effect that azithromycin is giving is quite small, if perhaps even negligible. So we have to wait for clinical trials to occur that are actually well performed and well controlled. But my guess is that if there's an effect, it has little to do with its antibiotic activity.
Thank you for that. It's amazing how quickly scientists are moving, and yet how we still have to be very patient. I'm always thinking about that as I hear new reports. The next question is really two questions, so I'm going to put them together. First one would be, what's the reason for the lack of interest by Big Pharma in new antibiotics discovery? And then a corollary question would be, in your opinion, how could Big Pharma be incentivized to look into new antibiotics? So let me run that through again for everybody. Part A, what's the reason for the lack of interest by Big Pharma in new antibiotic discovery? And Part B, in your opinion, how could Big Pharma be incentivized to look into new antibiotics?
Yeah, these are great questions. So the main, there's a couple of reasons why pharma has backed away from this. One of them, quite simply, is that antibiotics cure diseases. So if you go back and look at the kinds of medicines that are making the most money right now, cure disease, they usually control disease. So things like blood pressure medicine that you have to take for maybe the rest of your life, or cholesterol lowering agents, or anti-anxiety drugs. These are taken for long periods of time. Antibiotics are taken for a week, and then you get better, and with any luck, you'll never have to take it again. So it's quite obvious then that unless you charge a tremendous amount for that course of drugs, it's very hard to make your money back because you don't have a constant you know, consumer who's going to use this drug on a regular basis. So one of the things that happened in the golden age of antibiotics is that they realized that another consumer is the agricultural sector. And so pharma would sell the same drugs to farmers, and I mentioned oftentimes 10 times more, even 100 times more than the clinic. And so that was a great source of revenue. So you could have drugs that maybe didn't make tons of money for you, but you're making money in a different kind of way. That's all changed over the last 25 years, where most countries have really ramped down the use of antibiotics in agriculture, realizing that it's actually contributing to drug resistance. And so there's that component of it. They're just not profitable drugs. And the second component of it is that there is, and this is a little bit in the weeds, but a shift in the way that drug companies went after drugs in the 1990s. They used very large libraries, combinatorial libraries of chemistry or molecules. They used large robotic systems, and with the idea that you could just, interrogate millions of molecules against, thousands of molecular targets. And that has worked out very well for things like cancer, cardiovascular disease, but it's been an absolute failure for antibiotics. And almost all the antibiotics that we've discovered, in fact, all of them, have been discovered using what we call a phenotypic screening. You put a drug or a potential compound on on a susceptible pathogenic bacteria and see if it dies. That's not a great assay for these new kinds of automated systems. And so when they made that switch in the 90s, it made it hard to find new antibiotics. And so there's just failure after failure. It became very expensive to the drug companies as well. So some of it has to do with science, and a lot of it has to do with economics. In terms of incentives, there's a lot of activity out there to try and fix this problem right now. And there's a number of them. One idea is just simply to just guarantee drug companies a return on investment. So that's done, for example, with vaccines. Right? We will companies who are making vaccines with vaccine companies. They're going to buy, X millions of doses of their vaccine, and so they can be sure that their market is going to be there. And maybe we have to start thinking about things like that for antibiotics. The other idea, which I think is really creative, and it's being piloted in the UK in particular, and in some parts of Europe, is what's called the Netflix model. And basically what that means is government's subscription to a drug company or a consortia of drug companies to get antibiotics, to incentivize antibiotic discovery and to get antibiotics when they need them. And I think that model might be the one that might win overall. That is, it's more of a not-for-profit kind of model, but it guarantees a revenue stream for the folks who are taking the risk to be able to do these discoveries, because we desperately need
them. Thank you. That was really helpful. Jerry, there's a couple of related questions here about countries where antibiotics are easily accessible, very normal to get them from street vendors, so low and middle income countries, without proper regulation. How can that impact antibiotic resistance on a global scale? So it's referencing, there's two questions about where countries don't have regulations in place, antibiotics are easily accessible, whether from street vendors or wherever. Will that impact antibiotic resistance on a global scale? Your opinion on that?
Yeah, the evidence is very clear that it does, and it's no surprise that if you go back and look at those maps that I showed you, that for the most part, where you see the most drug-resistant organisms is in countries where that kind of practice is still up. It's takes some expertise to understand, like even in a modern, shiny hospital in Hamilton, oftentimes we don't know if a patient has a bacterial infection or a viral infection, for example. They have the same type, because we work on symptoms. I got pneumonia or pneumonia-like symptoms. Is it caused by a bacteria or is it caused by a virus? Without doing a test of some kind, it's impossible to know. And so even in the most modern public health systems, like in North America and Europe and Asia, we can't easily tell the difference between a viral infection and a bacterial infection many times. If you now transport that uncertainty into a country where they may not have the regulations and the public health infrastructure to help support it, and you couple that with ready availability of antibiotics, a series of collisions of opportunity for that the bacteria will thrive. Right? So even if, let's say you have a viral infection and you get an antibiotic from your pharmacist in one of these places, and I've been to many places around the world where it's very easy to just walk in and say, I've got a cough, and then they give you azithromycin, for example. Then if you don't really need that drug, then you end up with exposure, and your microbiome gets exposed to that. And of course, as you excrete that drug, the local area becomes contaminated without the sewage system and everything. So you end up with this perfect storm that is based on oftentimes a poor understanding of what's causing your infection. And if you couple it, as I said, with a lack of regulation as to what you can easily get to treat that, then you end up with a real mess. So, and it's been shown over and over again that a lot of the most resistant organisms that we find in the world right now are in these, or have their origins in these, where these practices are happening.
Okay, not good news there. I'm going to combine two more questions for you, Jerry. So I'm sorry, did I interrupt you?
No, you didn't. No, I was just reading you.
Okay. I'm going to combine two questions again, so hang on to your hat there. We're talking about two changes in terms of approaches. One was by the endorsement of the Global Health, the Global Action Plan on AMR by the WHO, and the other was the One Health approach to combating antibiotic resistance. So those two different particular programs. Have you seen a shift in R&D or a shift in combating the actual antibiotic resistance since those two particular initiatives have come to the fore? So that would be the Global Action Plan on AMR by the WHO and the One Health approach. Have you seen any changes or adjustments as a result of those two initiatives?
Well, with respect to the first one, the Global Action Plan, certainly what it has done is really become a rallying cry around the world for doing something, which, when I started in this field in the early 1990s, when as a postdoctoral fellow and when in Boston, where drug resistance was becoming a really significant issue and patients were failing their therapies, there was no understanding at all, or no in that this was a significant issue in policymakers. The Global Action Plan has served as a rallying cry for that, and all the members of the G20, G7 have agreed to develop action plans. Many of the other countries in the UN have done the same. The challenge in all of this has been funding them properly. So in Canada, for example, we're coming up with an action plan, but there isn't $1 underneath it. There's no directed funds, and so plans that have no resources are worse than useless, because they just waste everybody's time. And that's the situation, frankly, that we're in right now. I'm hopeful that that's going to change over time. And I think that this pandemic has really made people stand up and realize that infectious diseases can be incredibly difficult and to manage and can continue to be part of our lives. So I'm happy with the action plan. It makes a lot of sense. I'm disappointed with the uptake across the globe, frankly. There's been some places in the world, in Europe, for example, UK has been leading the way in this area. But even then, there's not enough resources to do this right. The other element that you asked about, which is the One Health angle, I think that's incredibly important. As I mentioned before, a lot of the antibiotics are used in agriculture and in particular raising animals to feed 6 billion people with limited resources. Understanding that antibiotic use in animals finds its way to humans either directly or through the environment, that triangle between environment, human health, and animal health is absolutely rock solid and backed up by great science. And it has been denied in many cases for decades by interest groups who want to continue to do this. I think it's really important that we all work together to try and find alternatives for antibiotic use. We can't because these kinds of practices tend, of course, to affect the poorest countries where we're coming out of, we're trying to feed a large population, we're trying to deal with a lot of a challenging infrastructure to be able to do that. So how do you do that safely? How do you do that without the use of antibiotics that imperils our use of them across the globe is something that requires a tremendous amount of attention. So the One Health idea, I think, really is bringing this to the fore, and I'm a huge fan of it.
Thanks very much, Jerry. That was really, really helpful to do those two in one. First, I want to thank all of our participants for those compelling questions. And we hope that the exchange with Dr. Wright was relevant and useful in expanding your understanding of pandemics. And I want to have a special thanks to our speaker, Dr. Jerry Wright, for an excellent presentation, and especially for the insights provided in the Q&A with our global audience. Jerry, we really appreciate your distinctive contribution to this webinar series. So let's quickly wrap up the webinar. Thank you again, all of you, for attending today's session. And we look forward to hosting the fifth webinar entitled Ethical Dimensions of Quarantines, Isolation Declarations, of States of Emergency, and Community Lockdowns. This will be held tomorrow, actually, Thursday, April 30th, from 10:45 to 11:45 Eastern Daylight Time. And it features Dr. Lisa Schwartz, the Arnold L. Johnson Chair in Healthcare Ethics, McMaster University. In this webinar, you'll learn more about the ethical aspects of measures to mitigate the spread of COVID-19. If you haven't registered for Webinar 5 or other webinars in the series yet, please register through the link on the website globalhealth.mcmaster.ca/2020 webinar series. So enjoy the rest of your day, everyone. We look forward to your participation in our next exciting webinar. Thank you. Merci. Gracias. Danke wel.